Hi, I'm Wayne Doyle. I manage the bioinformatics team at Active Motif, and today we're going to talk about the analysis of single-cell multiome, ATAC-Seq, and gene expression data. So over this talk, we're going to touch upon a couple of different things. First, what are the benefits of actually doing single-cell assays as opposed to our traditional bulk methods, and why do we want to do multiomics? And then I'm going to briefly go over 10x Genomics Multiome Kit, how the wet lab procedure works, and then go in detail about how we actually analyze single-cell multiomics data. So to start, I wanted to ground us in a biological question. So we're going to focus on leukemia. So leukemia is a blood-borne cancer, and it also has a heterogeneous disorder. So it's not just a single cancer in which there's just one group of bad cells. You have clones of leukemia cells, and this is important because those different clones have different genomes. There's been different mutations that they've acquired. They're expressing different genes. They have different transcriptomes. And then they also have different epigenomes. So their chromatin structure is different, where there's open chromatin, where there's closed chromatin. And this matters because if we're developing a drug treatment against leukemia, it may work really well against one of these subpopulations of leukemia cells. It will lower the other levels, but they're still circulating around, so you can get a relapse, which is not the goal that we're looking for when we develop a treatment for a cancer. So the question then is, how do we examine this heterogeneity and see what effect a treatment is actually having on the disease? So to start, I'm showing how we would traditionally answer this question. So this is an experimental design in which we have a control sample and we have a treated sample that we've given some type of drug. And then traditionally, we would do what are called bulk assays. So this is your traditional RNA sequencing, your traditional ATAC sequencing to look at chromatin accessibility. And this is where the researcher will collect cells, and then the lab will then process these as groups of cells. So we're looking across, on average, across all cells, what is the gene expression using RNA sequencing, or what is the chromatin accessibility using ATAC sequencing. We do this for the control samples, and then we also do this for the treated samples. And from this, we can get a lot of great information. This I'm showing here is just example data of an RNA sequencing experiment that we performed at Active Motif. There were 3 untreated samples and 3 samples that were treated with a compound. Each column here is a different sample. So we have our 3 replicates versus the 3 replicates. Each row is then a different gene. So if you have increasing red color, that means that gene is increasing expression. Increasing blue color means that gene is being decreased. So with this treatment, we can see there's several genes that are being upregulated in effect to the treatment. This top group of genes here, and there's also genes that in response to the treatment start to go down. So this is really informative. It allows us to get a first idea of what effect could my treatment actually be having on whatever disease or question that I'm trying to answer. But there is a limitation to this analysis because we're doing a bulk RNA-seq analysis. So we're looking on all the cells in the tissue or the sample on average, which means that if we have an increase in gene expression, that could be that every single cell that we're looking at had an increase in expression. And so your treatment's causing everything to go up. Or because we're looking on average, most cells could have had no effect. But if there's one subpopulation of cells that's responded very strongly to the drug treatment, for example, the red cells here, it's going to pull your average up. So it looks overall as if your average is increased when it's really due to just one cell. And you could interpret that incorrectly and think that everything is changing. Similarly, a decrease, that could be a decrease to all cells in the sample, or just one population really shut down its gene expression. And even when we see no change in gene expression, that could mean that we truly do have no change in gene expression. Or it could be one population of cells really increases gene expression, one population of cells really decreased it, so on average, we're not seeing an effect. And this limitation isn't specific to just bulk RNA sequencing. We can also see it with bulk ATAC sequencing. So ATAC sequencing is just an assay that allows us to look at regions of open chromatin. So these are regions where transcription factors can bind or RNA polymerase can come in to drive gene expression. On the left, this are browser tracks. So there are 4 samples, 2 within one group, 2 within another group. And then these peak looking things, these are sites of where there's open chromatins. We have reads that are accumulating at a region that was open in the samples when we did this experiment. And so we have some peaks that are open across all the samples. And then we also have peaks that are specific to one group. So here, there's decent peaks that then decrease. And then we also have peaks that are open in the second group. Very nice, strong signal, not present in these samples. And similar to the RNA sequencing data, we could either interpret this that all cells in response to the treatment start opening chromatin here, or there's just a specific subpopulation that opened its chromatin, pulling the average up. And the same for a decrease or for no change that we saw with the bulk RNA sequencing. So bulk assays are really great for getting us initial analysis. There's been a lot of really great science that's used them, but there is a limitation, especially when we're working with very heterogeneous samples, samples that have a lot of variability. We can never be 100% confident on what is actually responding to whatever our biological effect is. And so that's the benefit of the single cell approaches, which have been developing over a little over the past decade. And so in this, instead of looking at groups of cells, we now actually are profiling gene expression or chromatin accessibility within individual cells. So here we have the same experimental design that we had before. We have a control sample, we have a treated sample, we collect the cells, but now instead of processing all of the cells at once, we get gene expression using sRNA-Seq for each individual cell and chromatin accessibility from each individual cell using ATAC-Seq. One limitation with this, though, is that for this processing, we have split this sample. So some cells go to scRNA-Seq, some cells go to ATAC-Seq, but we're not directly profiling gene expression and chromatin accessibility from the exact same cell. So here I'm just showing some examples of some of the great results that you can get from these assays. On the left is a gene expression study on single cells in which they're looking at myeloid leukemia, and they're identifying groups of similar cells. Each dot here is an individual cell, and then they're colored by cells that have similar patterns of gene expression. And then in the circle are amyloid leukemia cells, and they're able to, based off the gene expression, determine what lineage these cells originated from. And so that can help inform how you actually want to treat these kinds of samples, or what experiments you can do based off their patterns of gene expression. And then on the right, I'm showing some chromatin accessibility data from ATAC-Seq. These are from PBMC cells. And we're looking specifically at enrichment for different transcription factor motifs, either CEB, PB, or GATA1. So these are transcription factors, so they bind to specific regions of the DNA that have very specific sequences. For example, they're shown here for the 2 transcription factors. And typically they'll bind to regions of open chromatin. So what we can do is look at all of our peaks of open chromatin and see if we have a relative enrichment for these specific motifs. So this increasing red color means that these groups of cells have open chromatin that could bind CEB, PB, or GATA1. So this is really great. We've started doing some really fantastic experiments into these that really help us really drill down into the biology of cells. But we have processed each of these separately, so there are still some limitations. We can get a pretty good approximation that the same cell types are found in each assay, but since we're not profiling gene expression and chromatin accessibility from the same cell, we can't directly say that a given chromatin accessibility profile is exactly matched to a given gene expression profile. And with these transcription factor data, we know that there are peaks that this transcription factor could bind to, but that doesn't necessarily mean that that transcription factor is actively doing something at this moment. It could be that those cells are primed, that if some type of stimulus turns on GATA1, then they can have a response, but it doesn't mean that GATA1 is doing something right now. And so that's why we really need to focus on moving into multiomics approaches, where we can look at multiple different assays together and get a whole picture of what is happening in a cell. So multiome is just a term for using multiple assays to probe different features in a biological space. So here I'm showing some of the different assays that Active Motif provides services for. And if we look at these assays to provide different information, so by doing RNA sequencing or single cell RNA-Seq, you can look at gene expression, get an idea of what the cell is currently doing. You can do RBS, look at methylation. You can get an idea on chromatin accessibility and chromatin conformation, what the 3D structure of the genome is, with a combination of Hi-C or ATAC-Seq. You can also look at histone marks and transcription factors, as well as proteins. But importantly, how the cell is behaving is not just one of these individual results. It's a combination of all of them together that drive the biology of this cell and how the cell is going to respond to some type of stimulus, be it a drug or something else. So in order to answer a biological question, it can often be very informative to combine two different orthogonal types of data sets together. And that's the benefit of 10x Genomics Multiome Kit, because it directly combines single-cell RNA-Seq to look at gene expression and single-cell ATAC-Seq to look at chromatin accessibility in the exact same cell. So now we're able to say, this is what my cell is doing in gene expression, and then this is how the chromatin accessibility is relating to that. So I want to briefly run over how the 10x Genomics procedure works. What we do is we first isolate nuclei, and then we do transposition in bulk. So transposition is just a term for where we add adapters to regions of open chromatin. This allows us in the downstream steps, we can amplify on those adapters and get reads from regions where the chromatin was accessible. And so once we've done this transposition, we run it through a machine in which each nuclei gets added to a droplet of oil. That oil also has a bead with the reagents that we need to do reverse transcription. So that means we can amplify up both the mRNA and the open chromatin on a single nucleus basis. And then since the nucleus is in a single droplet of oil, we're doing on a per nucleus amplification. And this amplification also adds what's called a barcode. These are just short sequences of nucleotides that allow us to say that anything that mRNA molecule or open chromatin reads that we obtained all came from this purple cell, and then there's a different barcode for this yellow cell. So when we do the downstream analysis, we can get information on a per cell basis. And then after we've finished the wet lab procedure, we then perform sequencing, where we will sequence the ATAC-Seq library and the gene expression library separately, and then we do a computational analysis to combine them. So here's an overview of how we actually analyze the data at Active Motif. It's divided into two phases. The first, we use a software called Cell Ranger. This is a great software package that was developed by 10x Genomics for use with their kits. It enables us to do a procedure called demultiplexing, which is just a procedure where we take the raw sequencing data and then we transform it into what are called FASTQ files, so we can perform analysis. It also performs a first pass analysis, so we can do clustering to identify some cell types. We can look at gene expression and also does some quality control steps to let us evaluate the project on a whole. And then it also pairs well with a software package called Loop, which allows you to do your own investigatory analysis of the data. And although Cell Ranger is really great, it gives you a really nice first pass analysis of the data. We also like to do a more in-depth analysis where we do more refined quality control, we do more refined clustering, and we do some more advanced analysis of the data. And so that's in the second phase of the analysis that we perform, where we use software packages developed by Rahul Satij's group, and they're really the gold standard for performing these types of analyses. And in each step of these, we have multiple quality control steps to ensure that we're passing along only the highest quality data and the highest quality analyses. So I'm not going to talk too much about the actual Cell Ranger steps. 10x Genomics offers a lot of very informative webinars about what the outputs of Cell Ranger are. They also offer events in order to meet with other people that are using the Cell Ranger software. So for now, and for this webinar, we're not going to touch upon Cell Ranger specifically, but instead focus on the second phase of the analysis that we do. So the first thing that we do is an additional quality control. because although we've already done some quality control through Cell Ranger, we want to make sure that any data set that we're looking at, we're not going to focus on just technical effects. We want to make sure that we're focusing on the actual biology so that we're answering the question that we want to answer. And there's a number of different metrics that we look at. On this slide, I'm only going to go over a couple of them. There's several more that we do as well when we're performing an analysis. So on this slide, I'm showing 3 different plots. These are violin plots. It's basically just a density plot that you've mirrored. And so on the y-axis, we're showing the number of some specific measurement that we're looking at. And then this plot then tells us that, for example, a lot of the cells have counts that are around this number of unique molecular identifiers or around this transcription start site enrichment. And then this allows you to visualize across your entire population of cells what's actually happening. And then on these gray boxes are the quality control thresholds that we actually used for the analysis. For most of our metrics, we determine this on a per experiment basis. So we make sure that we're giving you results that match your data as opposed to a rigid standard that could change from experimental design to experimental design. So there's a couple things that we're looking at on this plot. The first is the number of unique molecular identifiers. So this is just the number of unique reads that came from the gene expression data set. And the reason that we look at this is that during the wet lab procedure, a single nucleus is added to the droplet of oil that enables us to amplify up the mRNA molecules and the chromatin accessibility data. But no procedure is perfect, so there'll sometimes be a droplet of oil that has a lyse nuclei, so it just has kind of free-floating RNA in it, or you'll get a droplet of oil that has multiple nuclei in it. Both of these happen very rarely, but we don't want to base our interpretation of the biology based off something technical. Because if you have 2 nuclei together, you're not necessarily sure that those are the same cell type within it. They could be 2 different cell types, and then you could interpret that you've discovered some new novel cell type when really it's just a technical artifact. So we help identify these by first removing nuclei that have too few reads, because these are typically nuclei that have had some type of lysis or they're an empty droplet. And we also remove barcodes that have too many reads, because often if you are starting from 2 nuclei instead of 1, you have more starting material, so you typically have a lot more reads than the rest of the population. And then on the ATAC-Seq side, there's a couple things we look at. One of them is enrichment at transcription start sites. So in order for a gene to be expressed, its promoter needs to be accessible to RNA polymerase, and so that typically comes across as open chromatin. So if you look across all genes, you should see a really nice enrichment of chromatin accessibility at the transcription start site. And so this is a plot just showing that relative enrichment in which most of the cells are really nice open chromatin at promoters. And then the final plot that I'm showing here is we do a quality control based off the number of reads that we get from mitochondria. Some studies have shown that if you include cells with really high mitochondrial content after this wet lab procedure where we've already isolated the nuclei, that those can be sometimes lower quality cells. So we like to do a little bit of a selective cutoff here. We can adjust this though for experiments that have a highly energetic sample. We can change this threshold to better match what the data set is. And so we combine all the different metrics together and then only keep the barcodes that are associated with cells that pass all of our quality control. So we know that when we do the next analyses, we're focusing on something that's going to give really high quality results. And so the next step that we do is that we normalize the data. For this webinar, I'm not going to go into detail about this. On a conceptual level, we have thousands of cells that we're looking at. And so we don't want to base our biological interpretation from some cells having more reads by random chance and some cells having less reads by random chance. So similar to what we would do for a bulk assay, we just normalize the samples down so we can do fair comparisons from cell to cell. Or I'm going to focus primarily on for the next couple slides is where we do clustering. So clustering is just a procedure where we find groups of cells that have similar gene expression and similar chromatin accessibility, which means they're likely cells that are having a similar function within the body. So they're what are typically called as a cell type. Things are going to behave similarly in response to a treatment or in response to a stimulus. So before we can get to actually identifying cell types and clusters of cells, we have to do a little bit of data processing. So for each cell that we process, we're looking at 30,000 genes to see what the gene expression is, this row up here. And then we're also looking at, in many cases, more than 50,000 peaks and seeing what are the level of open chromatin within those identified peak regions. Which means that for a given cell, we're then looking at 30,000 features by 50,000 features, which is a lot of data to look at for just a single cell. But for these experiments, we're looking at thousands of cells, which means that our feature space that we're looking at is gigantic. It's not something that, as a human, that we can just look at and really get interpretable patterns from. We have to do analysis in the computer to figure out what the actual results are. And this can be large enough that if we're trying to analyze everything at once, it's going to take a prohibitively long time to run the analysis. It's going to use a lot of memory. It's going to use a lot of computing power. And we could also run into risks that we're going to overfit the data. So that's a situation in which you have kind of noisy gene expression just based off technical effects. And you base your interpretation of the biology based off those. So what we do is we just reduce the dimensionality of the data. And we do that by finding in the gene expression space variable genes. And so I'm showing here a number of genes that are expressed in a given cell. And we just say across the population of all the cells that we're looking at, what are the most variable genes? Which ones are having the most changes in expression across all of these cells? Because typically these are genes that are then playing some biological role, because there's a group of cells that are expressing the gene, there's another group of cells that aren't, so that leads to the appearance of high variability across all of the cells. And for the gene expression data, we just look at the top 2,000 genes. We can change this. We found that 2,000 genes often works well. This is well supported by the literature as well. And on the chromatin accessibility data, we get the top 95% of peaks. We drop the bottom 5% because typically these are a little bit noisy. They're more from technical artifacts and we don't want them to bias the results. But even when we're looking at 2,000 genes, that's still a lot of information for the thousands of cells that we're looking at. So I'm showing here a heat map. Each column here is an individual cell. Each row is a different gene. And then increasing yellow color means that there's increasing expression of that gene within that cell. And what we can see is that there's some noisiness. So you'll have cells that seem to be expressing similar patterns of gene expression, but then you'll see some of them have kind of this blue color. That could be biological. It could also be just something called a technical dropout. We just didn't happen to amplify that gene in that cell. So we want to avoid interpreting that too much in case it's coming from a technical artifact. And we also can see that there's a lot of these correlations. So we don't necessarily need to be looking at individual genes. We can instead look at groups of correlated genes and base the analysis off those. And that's actually the basis of something called PCA, principal component analysis. So that's a step that we do before we move on to the clustering. I'm not going to go through the math of PCA on this webinar. What we can functionally think that it's doing is this a mathematical operation that enables us to find groups of genes that are correlated with one another that help explain a large part of the total gene expression that's happening in the cells across all the population. So for example, the first principal component, there's a group of genes that are going up in one group of cells, a group of genes that are correlated and going down in that same group of cells. So we don't need to look at each of these genes individually. We can just get a single number that represents all of these genes together. The second principal component is then just a different group of correlated genes that helps explain a different part of the total gene expression that we want to look at. So if you combine enough principal components together, you can then fully recapitulate your entire gene expression space. But typically, if you look at just a small subset, we look at 50 by default. That typically allows you to capture most of what's happening with gene expression, but you're now looking at just 50 numbers instead of 2,000 numbers per cell. And we do something very similar for the ATAC-Seq data set as well. We do a procedure called singular value decomposition, otherwise known as SVD, that enables us to do a similar analysis using the peaks. And so for each of these principal components, they're each conveying information about gene expression, or singular values are conveying information about the chromatin accessibility. And so this plot is showing each dot is an individual cell, or plotting the first principal component versus the second. So cells that have similar patterns of gene expression are going to be next to each other, and cells with different patterns of gene expression are going to be further away. But we can't just look at two principal components, because there's still going to be a lot of very informative biology on the third, the fourth, and so on. So we would actually be looking at this data in 50 different dimensions for gene expression, 49 dimensions for the ATAC sequencing data. And so that's not something that we can visualize in our brains, because once we get past 3 dimensions, that's pretty much our limit. So in order to find cells that are similar to one another and having similar patterns of gene expression, similar patterns of chromatin accessibility, we do a procedure called a nearest neighbors graph that enables us to do clustering using a different mathematical approach. So I'm just going to talk about this on a very high level, in that we have a cell shown here in red. Based off both its gene expression and its peak profile, it seems to be very similar to this cell that's also colored in red. These two cells are similar to this cell, and then all of these red cells are similar to one another in both the gene expression and the chromatin accessibility. And similarly, these blue cells are similar to one another, but these blue cells are fairly different than these red cells or from these green cells. So we would then say this group of red cells is one cluster of cells. They're having similar biology. They're probably having a similar functional role in whatever tissue they originated from. And these blue cells are a different cluster of cells having a different functional role. And these clusters are often sometimes called cell types as well. So this is what we actually perform the analysis on, are these different clusters of cells. We look to see, are there different patterns of gene expression? Is there differences in the chromatin accessibility, and so on. To help us visualize it, we actually do a second procedure called UMAP embedding, which is uniform manifold approximation and projection. So this is a procedure where this neighbors graph is in however many dimensions we're looking at from the gene expression and the chromatin accessibility data. So we're basically just saying, if I tried to look at this big graph in 2 dimensions, what would it most look like? This is an approximation, so it's not ground truth. The truth is up in those high dimensions, but at least allows us to get a first pass visualization of the data. There are very valid criticisms of the UMAP embedding. You can't really interpret what distance means or what the shape of the clusters means, but it does allow you, that we'll see in a little bit, you can visualize what your chromatin accessibility is or what your gene expression is and see how that relates to groups of cells. So now that we've gotten our clusters, which is our focus of our analysis, we can start doing some of the fun, looking at what are the genes that are being expressed, how does that relate to chromatin accessibility, what are my potential markers, what are my differential genes. So we can start doing that now that we've really defined what are my groups of similar cells. So I'm showing 2 examples here from our standard analysis that we provide. On the left, this is that UMAP embedding, which instead of coloring by cluster, I've now colored by the gene expression. So each dot here is an individual cell. Increasing yellow color means that we're having increased expression of a gene. In this example, it is a gene called MS4A1. And so we see that this cluster here, cluster number 5, it has very strong expression in almost all of the cells for MS4A1, but we see very low expression in the other cells. So this would indicate that MS4A1 is a potential marker of this cell type. If we're doing downstream experiments, this would be a good target to focus on for manipulation. So that's really great from the gene expression space. We can also see on the chromatin accessibility space that the data matches. So I'm showing here, each of these lines is a different browser track. Similar to what we saw with the bulk ATAC sequencing data, if you're seeing increased signal, that means that there's increased open chromatin. We're applying this around the MS4A1 gene. So in cluster 5, the same cluster as here. We have really nice open chromatin at the promoter of MS4A1. This makes sense. We want the promoter to be open so RNA polymerase can come in and lead to the generation of the transcripts that we're detecting here. This violin plot here is actually showing the gene expression. So we have open chromatin and we have high gene expression. But something that's really important here is this bottom track that's in gray. It's a little bit hard to see, but it says all cells. So this is what's called a pseudobulk experiment. It's basically, we're saying if we had done bulk ATAC sequencing as opposed to doing single cell ATAC sequencing, what would the results would have looked like? So we would have gotten a very nice strong peak at MS4A1, which we expect because our single cells have that peak. But if we were looking at that data and we were just doing a bulk ATAC profiling, we may have interpreted, okay, I've isolated my sample. I'm seeing this really strong peak at MS4A1. That must mean that every cell in my sample has open chromatin at MS4A1. When in reality, it's just a subset, and based off the number of cells that we're working at, actually a fairly small subset of the total cells that are driving this open chromatin. And so that's one of the really strong benefits of using this multiomics technique, is that you can, one, pair your gene expression to your chromatin accessibility, and two, you can actually see the effect that individual small populations of cells can have on what you would get from an average experiment. And so that if this had been a drug treatment, we then may have misinterpreted it when it's really due to a specific cluster of cells that we're looking at. In addition to just pairing gene expression and chromatin accessibility, we can also do some other kinds of analyses as well. So here I'm showing enrichment of transcription factor motifs. Again, motifs are just sequences of DNA that transcription factors preferentially bind to. So we can look at these peaks of open chromatin across the cells and then see which motifs are found within these open called peaks of chromatin. And then we can say for each cell, does it have a relative enrichment for motifs being accessible for a given transcription factor? So here I'm showing for CEB, PB, this is actually going to be able to see on the next slide, two different clusters of cells. They both have very high expression accessibility for CEB, PB motif. So this indicates that this transcription factor potentially could be regulating both of these clusters of cells right now, and that it's being expressed is actively regulating gene expression. Or you can interpret this that these cells are primed, that if some stimulus turns on CB, PB, then that will lead to a change in gene expression. If we were doing single cell ATAC sequencing alone, that's kind of the extent that we can interpret it. But we were doing a multiome assay, which means we can now directly look and see, is CB, PB actually being expressed? And so that's what I'm showing here. On the left is just the same motif enrichment plot that we had on the previous slide. I've now circled it so you can see which is cluster 0, which is cluster 6. And on the right, I'm actually showing the gene expression. So it's a little bit hard to see on the UMAP embedding because we have a lot of cells that are on top of each other. So it's a little bit easier to see on these violin plots of the normalized gene expression. So cluster 0, this large cluster, there's some expression of CEB, PB. We can see that even here too. There's these nice green dots, but it's intermixed with these purple dots. So overall, it's not like every cell in cluster 0 is expressing this gene. It's just a subset of them. On the other hand, cluster 6 actually has very high expression of CEB, PB in most cells. So one interpretation that you get from this is that cluster 6, it's expressing this transcription factor. It has the motifs for this transcription factor accessible for it to bind. So CEB, PB is probably actively having a regulatory role in this cluster. An interpretation of this data for cluster 0, which has lower expression, is that this could be cells that are primed, that if some type of stimulus turns on CEB, PB, it can then have a very rapid effect. All of its sites that it needs to bind to are sitting there ready for it to be expressed. It just needs that gene to turn on. And so that's two different interpretations that we wouldn't have been able to get if we had been looking at this data on the gene expression alone or the chromatin accessibility data alone. And to help us identify kind of what we should be focusing on, We also do differential analyses. So we identify differentially expressed genes between clusters or differentially enriched peaks between clusters. So I'm showing here an example. There's a couple genes, cluster 1 and in cluster 2, and then we want to do a test to see, is it statistically significant that, for example, cluster 2 has high levels of KIT, and that they then don't have very high levels of differences in CBPA, for example. This is just toy data. It's not biological in this example. So we do automatic statistical tests that enable us to determine the fold change enrichment. So we can see, does a specific cluster have high levels of a specific gene compared to the rest of the cells? We give you the normalized expressions. You can see overall, is it really highly expressed in that cluster? Is it lowly expressed? Then we also give an associated significance for how much it is enriched. So these are all genes that are very highly enriched in the cluster 1 that we analyzed. And then we also do this for the ATAC-Seq data. We'll look at differentially accessible regions and see if they're preferentially open or closed in a cluster. And we'll also look to see if specific transcription factor motifs are specifically being enriched or decreasing in clusters, which enables you to get a very nice overview of the entire data set. And then we also plot these differential genes, so we get the top ones and then put them on a heat map that enables us to get a quick overview of what does my data set look like, what are the marker genes that might be identifying my specific clusters. That way, if you know from the literature or from other experiments that you've done that I know that one of my cell types always expresses a specific gene, you can then look at this table and see if that gene's present and which cluster has it highly expressed. So this can be really used as a foundation to start doing some cell type annotation. Similarly, the tables that we provide can also do that as well. This is just a nice visual representation of it. And what's nice also, since we're doing a multiome assay, is we can identify differentially expressed genes, and then we can match that up to the differentially accessible peaks. So I'm showing here CD6 was specifically enriched in cluster 1. See that it has fairly high normalized expression. It's relatively enriched, it has a strong p-value. And then there's a peak that was nearby, close to CD6, that is also statistically enriched. So this is a potential gene regulatory element that is opened in this gene, and then correspondingly, the gene expression is also increased. So this is a potential target for manipulation if you want to modify the expression of CD6. So that's a brief overview of the multiome procedure and how we analyze it here at Active Motif. And just to summarize, what we really gained from the single cell multiomics approach is that as opposed to some of the other assays that we do, in which we're looking at chromatin accessibility alone or gene expression alone, we're now defining cell types based off orthologous data. So this allows us to get a much more robust interpretation of what the actual identity of these cells are. And so showing here, these are just different plots. They have the same clustering, but this is a plot based off the chromatin accessibility, a plot based off the RNA sequencing, then a plot based off the combination of them together. And so you can see we get a little bit more refinement when we look at both data sets together. And then also from this type of assay, we get both our gene markers, but we also get the regulatory elements linked to them. So we can start teasing apart, how is gene regulation playing? Where are the potential motifs that are driving the identity of my cells? And how does that relate to the gene expression changes that I'm seeing? And so that's one of the really great benefits of this assay. And it's something that is a strong benefit for moving into multiomics overall. All right, that's the extent of my end, Mason.